Hello, everyone, and thanks for joining us for the Landscape of Cancer Research Advances in Immuno-oncology 2024 online symposium. My name is George Hammond, and I'm excited to be here to moderate today's event alongside my colleagues Princess Sennett and Heather James. Before we start the talks today, I just have some general information and a few tips for getting the most out of the event. We would first of all like to ask if you could please refrain from video, audio, or photo recording of all talks. Many of our speakers have kindly agreed to share their slides with you, and these are available within the literature section on the platform. Please remember that we encourage debate and discussion and welcome all of you to use our networking services to introduce yourselves to other attendees, but we do not permit the overt solicitation of products or services and reserve the right to block any user who infringes on this rule. It's also incredibly important to say that this event was only made possible thanks to the generosity of our sponsors and partners, particularly our Technology Spotlight sponsors, Biomodal and Biorad. For more detailed instructions on how to make the most of the features available to you at this event, please navigate to the event home and watch the overview video at the top of the page or visit the FAQ section in the left-hand menu. And with that out of the way, I will now hand over to Heather to introduce our keynote session.
Thank you, George. With that out of the way, I will now move on to introduce our opening and keynote session for the event. I'm pleased to welcome Professor Sebastian Kobold, presenting Engineering Immune Cells for Cancer Treatment. Professor Kobold is Professor for Experimental Immuno-oncology, Head of the Immunopharmacology Group, and Deputy Director of the Division of Clinical Pharmacology at the University Hospital in Munich. He has received, among many other awards, the Cancer Research Award 2022 of the Berlin-Brandenburg Academy of Sciences, and the Georges Köhler Award 2023 of the German Society of Immunology. Focusing on tumour immunology, Professor Kobold is especially interested in developing and testing novel strategies to treat cancer using the immune system. A warm welcome to you, Professor Kobold. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. Without further ado, I will now hand over to Professor Kobold to begin his presentation.
Thank you very much for the kind introduction. A warm welcome also to my side. It's my pleasure to introduce you to a topic that is dear to my heart and has been the focus of a lot of the research that has been undergone in my group and my team over the last decade. And as introduced already, it will be about engineering immune cells more specifically, and for the topic, for the sake of today, about engineering T cells. So these are my disclosures, although I won't talk about them today. I think the only thing I need to mention here, for the sake of transparency, that I will be mentioning one clinical study of Adaptimmune, which by some may be seen as a conflict, but obviously I wasn't involved in that study. So as I said, it's the topic of today is engineering immune cells. And the purpose of that is to use immune cells for treatment of patients affected by cancer. And this is also, a more global strategy known or sometimes referred to as adoptive cellular therapy or adoptive cell transfer. And the topic or the name comes kind of obvious because what this is about transferring therapeutic cells to patients. And historically, this has come in two flavors. Based on the recognition that cancer-specific cells, T cells, exist in cancer patients, when it's come about over 30 years ago now, with the idea that these could be utilized for cancer treatment. And because these cells were thought to be most abundant at the cancer site, they were initially coined as tumor-infiltrating lymphocytes. That's what you see here. So these cells can be isolated from the patient's own tumor, expanded massively in the cell culture, and then reinfused back to the patient in therapeutic intention. As you can imagine, this type of therapy is cumbersome because you need to resect the tumor, to cut it out, to work with the tumor and isolate cells from it. Many patients may simply not have these cells, or it's difficult to know what they're doing, what they're specific for. And so it was very difficult to standardize this type of treatment. And we really waited for a very long time until we saw, and I'll come back to that in a few moments, clinical studies that would really demonstrate efficacy of this type of treatment in a randomized fashion. But I'll come back to that in a minute. And that's why very early on, researchers have envisioned that perhaps cancer specificity could be achieved by other means. It could be achieved by means of genetic engineering of the same cell, of the patient cell. And historically, again, so this has come in two flavors. And of course, you could divide this into many more flavors today, but that's not the topic. Either in engineering these cells with a specific T cell receptor of your choosing, that's specific for a cancer-associated antigen, or with synthetic receptor, and we'll come back to that, what these receptors are, also coined as chimeric antigen receptors, or CAR, to mediate therapeutic specificity against the cancer cell, and again, to use these cells engineered by this way to treat patients. So the rough differences between those strategies, so on the left side, the T cell receptor, which is the natural way how T cells sense the antigen in the target, which is a very complicated receptor, where the alpha and the beta chain pair to mediate specificity towards the peptide in the context of HLA, and recruit the other 6 chains, here the epsilon, the gamma, the delta, and the zeta chain, to assemble the full T cell receptor. And that may additionally require co-receptors like CD8 or others. In contrast, the CAR, or chimeric antigen receptor, is a much simpler receptor. It's a polyprotein constituted, as the name intends, by pieces of different proteins. It's a receptor that as such does not occur in nature, because you won't find any such chimeric receptor that is constituted of so many different pieces from different proteins. In the extracellular space, that's here, you have the minimal variable fragment of an antibody, a so-called single-chain variable fragment that mediates specificity. You have something that spaces it to the membrane, something that anchors it to the membrane, and intracellular you have stimulatory domains that initially were meant to mimic T cell receptor activity. This is why you find here the CS3 zeta chain, and also some mean of T cell co-stimulation. A major difference, additional to the structure, between both types of receptors is what they actually see and how they see it. On the right side, what you have is the way T cell receptors naturally sense the antigen, which is an antigen, a fraction of an antigen presented in the context of major histochromatic complex 1, MHC, on the surface of, on our setting, of a tumor cell, which is a reflection of what's going on in the cell. So in principle, it's a fragment of an intracellular protein, of a surface protein, or even of a virus that is presented in this context, and that mediates recognition of the T cell. So meaning that in principle, a T cell receptor can recognize also a broad repertoire of antigens, including intracellular antigens. In contrast, a CAR is restricted to what it sees on the surface. So it's restricted to surface antigen, which by nature are simply much less abundant than intracellular and surface antigens. So meaning that in a nutshell, you restrict yourself with CARs to a few thousand antigens that are present on the cell surface. So that's a big difference between these two types of receptors. But the CAR has a big advantage over the T cell receptor. It is HLA unrestricted. So meaning whatever your HLA type, you will qualify for this treatment if you have the antigen expressed on the cancer cell surface. Then there are also like some, more functional differences between both types of receptors, and we try to schematize it here. So on the top you have the CAR, on the bottom you have the T cell receptor. So typically the onset of a response with a CAR, because it assembles all these pieces in one receptor for signaling, is more rapid than for a T cell receptor. That's what we try to depict here. So you have the activation of the CAR, the degranulation that happens. Both of it happens earlier than for a T cell receptor, which has a slower onset, as you can see on the bottom. Then, because this happens faster, then of course the detachment of the cell from the target cell is of course faster as well compared to the T cell receptor, and then this can be repeated. So you also see that even at this very high level overview of how this receptor functions, you can also already have substantial differences between both types of receptors. The way they will ultimately kill is in fact very similar. Both will lead to degranulation of the cell, so meaning releasing cytotoxic cargo like granzymes and perforins. It also releases cytokine that can be cytotoxic or modulatory like interferon gamma. And also both come with membrane-bound death receptors that they upregulate, and that can also lead to death and targeting of the cancer cell, including antigen-negative cancer cells. And these mechanisms are certainly shared between both types of therapeutic cells, and sometimes can be more preponderant in a given CAR or T cell receptor type. The problem, or one problem with both types of therapy actually, is that because of this degranulation effect, and importantly also of cytokine production, is that once the CAR is activated because it recognizes antigen, it triggers an inflammatory response, which is desired to a certain extent, but at the same time leads to the activation of accessory cells locally and systemically, which can be desired up to a certain extent, but can also lead to an autonomous effect in terms of immune activation that mainly hits macrophages and potentially also other immune cells, which then can perpetuate itself independently of the CAR. This is a phenomenon also often described as cytokine release syndrome, which can be a very dramatic and also very severe side effect of CAR, also T-cell receptor therapies. And the bad thing about that is that it can in principle happen anywhere and at any time within the time frame of the first hours, days, and sometimes a few weeks upon treatment. The good thing is, of course, that once this has happened and has gone away, it likely won't come back, because this type of cellular therapies is a one-off treatment, and so it hits hard early on, and afterwards, once the side effects are clear, typically these types of side effects don't come back. But if they come, they can be very severe. And thankfully, because we have learned to treat and recognize these side effects also early on in the clinics, adequate management strategies these days allow to mitigate most of these side effects to a manageable size and treatment depth without compromising the treatment efficacy of the CAR T cells mostly. So again, this is another way of depicting how these CARs look, but it's the very same structure that you've seen before. And the purpose of this slide is to show you the 6 approved CAR product that we have right now. 4 of them are targeting CD19 for the treatment of acute lymphocytic leukemia of the B-cell type or B-cell non-Hodgkin's lymphoma. And 2 are targeting the BCMA, which is a plasma cell-associated antigen, which is found abundantly on multiple myeloma cells. So both antigens are the 2 only antigens approved, or where the CARs exist that are approved to treat diseases that are positive for these antigens. And just to give you a ballpark of what we're talking about here, so these are the 2 initial indications where CAR were approved: acute lymphocytic leukemia in adults and children, and diffuse large B-cell lymphoma. And what the numbers that are shown here is a summary across studies of the complete remission rates upon a single infusion of a CAR T-cell product. And you can appreciate that these numbers can range up to the 90% in acute lymphocytic leukemia, but sadly, many patients, if not most patients, will eventually relapse. And so meaning that in the end, about less than half of the patients that initially respond will stay in complete remission on the long run, but still, this would have been otherwise incurable patients. And in diffuse large B-cell lymphoma, the numbers of complete responders are typically lower, so in the range of 40 to 50% typically. But these responses, once they occur in the sense of a complete response, typically last in these patients. So meaning that these patients have a high chance of actually being cured from the disease, which is quite remarkable for an out-treated disease. So this is the story so far, the story of clinical application of CARs. But the story is not over yet. I mean, the landscape is changing rapidly and dramatically. So historically, of course, most studies, and that's what's shown here, about 2/3 in this study or this survey that we conducted almost 2 years ago now, 2/3 of these studies were in hematology. So it's not a surprise. That's where these CARs are most effective. But a growing now, at the time, third, and now it's certainly closing on more than 40%, were in solid oncology, and indicating that we're shifting the gears from bringing these CARs to these engineered cells to hematology, from bringing them hopefully one day to oncology. And this is a matter of very active ongoing clinical and preclinical research. And the reason why there is hope and belief that this might actually be a good way to go also to treat solid cancer, not only hematological cancers, is given among others by this study. So I referred to you earlier about this tumor-infiltrating lymphocytes and said it took a long time really to get this to the clinic, and it's partly to understand what the response rate of that was. And finally, 2 years ago now almost, the phase 3 clinical trial was published that randomized these tumor-infiltrating lymphocytes in malignant melanoma against the standard of care at the time, which was a checkpoint blocker in first-line checkpoint blocker refractory patients. And you can appreciate that we have a very clear pattern of response, including about a third of these patients that react with a complete remission of long-term stable disease. So indicating that with a single infusion of this product, this patient may be cured. And again, so this is a solid cancer, and this is the first evidence ever that that the solid cancer can be treated with a solid with a cell therapy against the very same cancer cells. And unsurprisingly, marketing authorization has been requested in Europe, and actually for a different TIL type of product, marketing authorization was already granted by the FDA in the United States for the treatment of malignant melanoma. But and that's not the only sign of activity that we have up to now. So as I said before, so this is sort of my potential conflict of interest. So this is a phase 2/3 clinical trial published by or conducted by Adaptimmune, which is a clinical stage company dealing with T-cell receptors. Here, T-cell receptor targeting MAGE-A4, so it's a cancer-associated antigen in synovial sarcoma. And you can appreciate that here we have a large proportion of patients that respond. And these responses are also durable, as you can see here in the spider plots down there. So meaning that the patient that undergo into a certain depth of remission stay at least for a certain time in remission. This is very meaningful in such patients that haven't seen any, advance in therapeutics for the last 4 or so decades, which is quite dramatic and can be quite meaningful to these patients. And I understand that the company has also requested marketing authorization, at least in the United States. And so it should be decided on that by this summer. The problem is that outside of the few indications, and even for the patient that we just discussed, we clearly have a large amount of resistances to cell therapies, although we know for a long time that you can infuse large amount of these cells safely into patients. But the problem is, also probably in these trials, especially in the patient that you don't see responses, is that these cells, most of these cells actually don't even enter the tissue. So meaning that how can a therapeutic agent be active if it doesn't reach its intended therapeutic target, which is in the cancer tissue? Second, of the few cells that eventually make it there, what we also see is that in solid oncology, it's a big difference to hematology. We have a large disease heterogeneity also in a patient and in a tumor site, where different cells would express different patterns of antigens, and thereby leading to an insufficient tumor cell recognition, because if you hit one antigen, perhaps it may be lost and others might become a preponderant, or the antigen was never consistently expressed in the first place. And last but certainly not least, we have the problem of local but also systemic immune suppression, which we know is very prominent in cancer patients and also impedes largely our therapeutic cells. And ultimately, we and many others believe that you will need to tackle all these aspects in one therapeutic product to deliver the full benefit of CARs or cellular therapies to cancer patients. So now I want to show you a few approaches that we've undertaken in my lab to tackle these issues and that we believe might be of clinical value. So how to move these cells into tumors? So one observation that we made, and this is kind of a textbook knowledge, is that immune cells follow certain signals to enter tissues. So these signals are called chemokines. It's soluble mediators or mediators of attraction. And the T cells or the immune cells are able to sense these if they express the matching receptor. And only then would they be able to follow them to enter tissue. So this is also a physiological mechanism of immune regulation. The problem is that in cancer, typically what happens is that the expression of this mediator is dissimilar or is not matching the expression of the matching receptor on T cells. So meaning these T cells don't have the mechanism to enter these tissues. And we hypothesized that if we could restore this matching between the T cell expressing a chemokine receptor and the chemokine expression in the tumor tissue, we might be able to drag these T cells back into tumors. So what we did with a collaborator of mine, Thorsten Mempel at MGH in Boston, so we scrutinized the expression of chemokine and chemokine receptors on tumor-infiltrating lymphocytes. And what we found is that the CXC motif receptor 6 was abundantly expressed in T cells infiltrating melanoma tissue. And so what we found out in the study here, that's just a summary, that this receptor is critical to attract T cells to sites, to distinct sites around the tumor that contain certain types of dendritic cells. And the purpose of doing that is that this contact with the dendritic cell would provide the T cell with essential survival signals by allowing them to see IL-15 presented in trans by IL-15 receptor alpha expression on these dendritic cells. And only when that happens would the T cells be able to proliferate and survive in a tumor environment when they're cancer specific. And otherwise, they would just die off and thereby not being able to mediate any functionality. So we hypothesized, based on these observations, that perhaps if we would arm therapeutic cells, especially CAR T cells, with this receptor, we might be able to drag them into tumor tissue and thereby mediate enhanced therapeutic potential. And this is just a snapshot of what we found. So, of course, we showed that this is exactly what happens. The cells are attracted more deeply into the tissue to perform the job better. And this is the consequence of it in two xenograft models depicted here. So the CAR would mediate some level of therapeutic efficacy, but many of these mice would eventually have to be taken out of the experiment. Whereas in the control, whereas in the group armed co-equipped with the CXCR6 receptor, we can see in both models complete rejections and survival long-term of the mice treated with this advanced engineer CAR T-cell product for augmented recruitment. I think this audience is well aware of the relevance of the PD-1 programmed receptor 1 programmed receptor ligand 1 axis in suppressing T-cell function. And there are a lot of agents that target this axis as a monotherapy to unleash the patient's own T-cells. So with a decade ago, with that in mind, so we envisioned that perhaps we could also use this signal to confer the T cell with additional stimulation. So turning the suppression that this receptor mediates into stimulation. And we designed PD-1 CD28 fusion receptors that would transform PD-L1 signal into co-stimulation. And we teamed up with a company at the time, which was called TCR Squared, to investigate whether their T cell receptor platform would benefit or stand to benefit from this PD-1 CD28 fusion receptors or switch receptors. And we demonstrated that here in a mesothelioma model that while the T cell receptors are effective, at least for a certain time, in controlling the disease, adding this boost to the T cell mediates more prolonged and sustained remissions in these mice, and importantly, leads to tumor rejection in most mice that we've treated, a durable tumor rejection in most mice that we treated, indicating that this might actually really do a difference. And this led to a phase 1/2 clinical trial, which is currently ongoing, combining these two types of receptors in patients that are positive for mesothelioma. The other problem that I've hinted at is that because of the heterogeneity of the disease, you need to find adequate targets that you could use to hit the tumor cell, which ideally would be so important to the disease that they might not be lost along the way. And to do that, so we established a novel methodology using single-cell RNA sequencing of whole tumors that would then allow us, of course, to distinguish between malignant and normal cells and to understand which cells would express what. We took here advantage of already archived, public single-cell RNA sequencing data sets, in this case as a pilot indication of acute myeloid leukemia, but we're doing this for many more diseases now. And use this to understand where the antigens that might be of interest for CAR T-cell therapy are expressed, and importantly, to identify such antigen that might allow us to differentiate between the healthy compartment and the diseased compartment. To benchmark this, we used a healthy atlas of the whole body. So using cells from healthy organs across the human body, at that time of over 500,000 cells, now we have more than 5 million of these cells across the human body, to be able to really understand where our targets of interest might be expressed. We benchmarked the criteria against all the targets that have been described up to this day for this disease. So one important criteria was, of course, the overexpression compared to hematopoietic stem cells. We needed cell surface antigens. We didn't want antigen to be on T cells because otherwise we would target ourselves. Importantly, we did not want to have this on any critical cell types across the body for safety reasons. And we benchmarked this in these different targets that were described for the disease before, and you can appreciate that none of these antigens actually fulfills this criteria, and importantly, not even the overexpression criteria. And what we found actually, by using this analysis, we find two new targets, which is CSF1R, so colony stimulating factor 1 receptor, and CD86 that were specific or selective, not specifically, but selectively expressed in AML compared to the hematopoietic stem cell compartment. That could be efficiently used to target AML blasts. And this is a snapshot of what we did here in a patient-derived xenograft model, so of an acute myeloid leukemia, injected into mice. And you can appreciate that this is typically needs to sacrificing the mice because of signs of disease relatively rapidly. Whereas our controls, our novel CARs targeting CSF1R or CD86 were able actually to clear the disease entirely in the mice stayed tumor-free for as long as we followed them up. So indicating that these antigens might be therapeutically very attractive antigens, and that perhaps this might allow for better and more selective treatment of acute myeloid leukemia. And we're currently trying to move this towards the clinic, and we're hoping that this might be actually a strategy that would allow for safer and better therapy of acute myeloid leukemia. So with that, I'm already at the end. I hope I could introduce to you that actually cellular therapies, importantly in the form of CAR T-cell therapies, are already clinically approved, at least for CD19 and BCMA positive diseases. And I think they're there to stay. And we are very curious to see if this portfolio can be extended, even in hematology. There is an emerging and growing evidence, and I've showed you two clinical trials, one phase 3 and one phase 2 trial, to indicate that there is evidence now for activity of cell therapeutic product, in this case of TILs and T-cell receptor engineered T cells in different solid tumors, namely, for the sake of today, it was about melanoma and synovial sarcoma. To allow broader application of cellular therapies with increased relevance, we will need to tackle the current limitations of cellular therapies, which, as indicated, are the access of the therapeutic cells to the tumor tissue, the choice of antigen or the combination of antigens for adequate and robust tumor targeting, and of course, local but also systemic immune suppression that is impeding the activity or the therapeutic activity of T cells in tumors, especially in solid tumors. I need to thank the different collaborators that helped us to generate the data that I've highlighted to you today. Of course, my team, my dedicated team here in Munich, who lives to make cell therapy work in different oncology and perhaps also non-oncological indication one day. Our different funding agencies that support us in our effort, importantly, the Collaborative Research Center of the German Research Foundation, Let's Immune, and the European Research Council. And with that, I'm at the end, and I would be happy to take any questions you may have.
Wonderful. Thank you so much, Professor Kobold, for that really fascinating talk. And as with all the talks, we're likely going to have lots of questions from the audience, and I can see that we do have some questions already, so I'll move straight into the Q&A. So our first one, someone has asked if you can clarify that difference between a CAR and a TCR for us, please.
Sure. So I think there are two layers of difference in essence. The first layer comes from the receptor itself. Although a CAR aims at, in a way, mimicking a T-cell receptor, because it utilizes similar, at least one of the similar signaling moieties, which is the CD3 zeta chain. Signaling is different between both, because the T-cell receptor, as you may have seen, is constituted of a total of 8 protein chains anchored in the membrane. The alpha and the beta chain that mediates the recognition of the target antigen, the delta, the gamma chain, 2 epsilon chain, and 2 zeta chains, which need to assemble. And all of these chains, but for the alpha and the beta chain, can signal. So meaning that in principle, with the T-cell receptor, you get a much broader range of signaling activity. But again, the difference is that while the CAR is already containing a stimulatory domain, the T-cell, co-stimulatory domain, sorry, the T-cell receptor doesn't. So meaning the T-cell receptor would need additional co-stimulation coming from somewhere else, either naturally or exogenously. So that's about the receptor. The other main difference between these two types of receptors is what they see. As I said, or I tried to explain, the T-cell receptor is restricted to whatever is presented in the context of the major histocompatibility complex on the surface of a cell. So meaning that if the peptide fragment that the T-cell receptor is specific for is not in the cell, then it won't see the cell. Or if it doesn't get to the MHC, then it won't see it either. But the big advantage is that, of course, in principle, for T-cell receptor, you can also target intracellular antigens, which by nature is a much bigger pool than what the CAR can see, which is only restricted to surface antigens, which are only a few thousand potential antigens that would even qualify for that. But of course, again, the big disadvantage of the T-cell receptor is the HLA restriction, because you can't use HLA, I don't know, HLA-A2 restricted T-cell receptor for someone that is restricted with HLA-A1 also. These are simply two different types of HLA and different restrictions. Whereas the CAR by nature is of course independent of such HLA type, and this is not limited to patients of the same HLA.
Excellent. Thank you so much for the clarity on that. Another question about the T cell receptors. What is the status of development of T cell receptor approaches?
Right. I mean, I think although of course the idea is decades old, and certainly as old as the T cell receptor itself, or at least as old as the CAR are, and historically. But the problem is that because of this complexity of T cell receptors, including so the structure, including the target profile, including HLA restriction, it's much more tedious. It was much more tedious to develop these approaches compared to a CAR, which is simply a one-fits-all type of approach, right? I mean, if you have the CAR, you can, in principle, introduce it into any patient cells and use it to treat the patient. Of course, there are more limitations to that, but just in principle, from the process, that's it. For T-cell receptors, you need the HLA typing of your patient. This needs to match. The patient needs to have the antigen you want to target. This needs to match also with the HLA type, etc. So that's much more complicated. But thankfully, I mean, because so obviously it just made a lot of sense to continue on this track. People have continued to study and to develop T-cell receptors. And as I tried to allude to, I mean, I think we have the first T-cell receptor, which is targeting MAGE-A4, on the verge of approval. We'll see, I think, about this summer whether this comes true for the treatment of synovial sarcoma. And this would clearly also be a breakthrough for this type of treatment, because it's the first cell therapy ever using a T-cell receptor that would be approved.
Excellent. Thank you for that. That sounds really promising, and I'm wishing you the best of luck. I'm sure that all the viewers are as well on that one. So another question that we've got is, how frequent are mutations in surface proteins on cancer cells?
Yeah, I mean, I think that's sadly, I would say, not as often as, you know, as oncologists or drug developers we would wish for. Because in fact, although mutations, as we all know, in cancer is a very frequent phenomenon, but these mutations mostly concern intracellular proteins, or at least the signaling moieties of extracellular proteins. So meaning that most mutations don't see the cell surface, which is of course a limitation to what we want to do, for instance, to develop a CAR against it, to develop an antibody against it. But there are a few examples. I'll just give you two that we worked on. One is the epithelial growth factor receptor variant 3, which is one of the very few mutated cancer-associated antigen that exists, which are mostly found on glioblastoma. The problem is to them, and that's at least what clinical evidence has suggested so far, is that this antigen, because it's not essential, it doesn't seem to be essential to the cancer cell, can get lost upon treatment pressure. And that's actually what happened in a clinical trial investigating CARs against this antigen. In contrast, we also have another type of mutated antigens, which is more functional to the cell, which is actually the B-cell receptor. The B-cell receptor on malignant chronic lymphocytic leukemia cell is something that actually has a function in the disease biology for certain of these patients, because a mechanism is that this B-cell receptor will actually recognize itself and thereby trigger cell autonomously proliferation and survival of these cells, of these malignant cells. And so that's and actually the mutations that occur in this B-cell receptor that trigger this mechanism are actually quite conserved across patients, because it's just a motif that is often recognized. And we at least believe that this may be a very attractive target for therapy, and actually developed CARs against that, and recently published this result, and hoping to be able to move that with our colleagues in Basel towards a clinical trial in the next few years.
Lovely. Thank you so much. That was really interesting. I think that we've got one more question that we've got time for.
Mhm.
And this one's really interesting here. Are there other ways to enhance access to cancer tissues that are not mediated by engineering?
um, so yes, I mean I think there are certain. I mean, let's put it that way. So I think there are two ways of contemplating that. The one way is, I would call it the rough way, because it's doing that, but it's not necessarily specific or tailored. It's what we call preconditioning or non-myeloablative chemotherapy, or it could be also done by radiation. But and what happens is that you get a relatively high dose of chemo, which may impact a bit your cancer, but typically not a lot. But what it does, it kind of like dampens your immune system. It causes tissue damage, which releases certain signaling factors, etc. And one of the consequences is that it stimulates cells, including the cells you would have transferred afterwards, your therapeutic cells, and it permeabilizes, at least very transiently, vessels, lymphatics, for cells to evade the bloodstream to the tissue more easily. So that's a rough way. There may be more targeted approaches like local therapies that could be also using radiation, could be using sometimes of thermal or cryoablation, that where you would apply these things locally to let's say a tumor tissue, or and thereby enhance the ability of these cells, of the therapeutic cells, to enter and access the tumor tissue. But this part, so the later part, the more local part, is certainly a matter of ongoing investigation, to which level this can actually really be used to trigger a CAR or therapeutic cell.
Excellent. Thank you so much for catching those questions for us. Unfortunately, that is all that we have time for today. So, any questions that we didn't have time to get to will be sent to our speakers after the event. And as with all of the other talks in this event, it will now be available to view whenever you want for the next 30 days. And that just leaves me to say thank you so much again, Professor Kobold, and we'll be back soon with the next talk.
Thank you.